All right, I think some people still join a little bit later, but I think we can slowly start. Welcome everybody to the Pages Early Career Network North America Show and Tell series. My name is Sandra Brücker. I'm one of the North America representatives and a postdoc at the Desert Research Institute in Reno, Nevada. Thank you all for attending today's Show and Tell event. Today, we are joined by Stella Mosher and Dr. Nathan Chellman to learn more about paleofire research in the South African Fynbos region and how we can measure black carbon in lake sediments. First, Stella will give us an introduction to the study area, and then Nathan will walk us through the black carbon measurement techniques. Stella received her undergrad degree in geology from the University of Puget Sound, her master's degree in geology from the University of Cincinnati, and she's now about to start her fourth year as a PhD student in the Department of Geography at the University of Utah. Stella's research focuses on high-resolution fire history in the South African Fynbos biome. She uses macro charcoal particles in lake sediment archives over the past 4,000 years to build fire histories at ecological scale resolution over millennia. Stella is particularly interested in the degree to which she can detect impacts on the fire regime related to the expansion of pastoralism around 2000 before present. She's also an adjunct faculty in the Department of Natural Sciences at Salt Lake Community College, and she's the proud mom of two orange cats named Bunsen and Beaker. As always in our Show and Tell series, you can write your questions in the chat box or raise your hand and then unmute yourself and directly ask your questions. And with that, Stella, I would like to give the floor to you for the introduction to this exciting Show and Tell event.
Great, thank you, Sandra. Let me just share my screen. All right. Okay, so just to introduce a little bit about the area and study site that we're looking at. This is a map of South Africa, and the purple section is South Africa's Fynbos biome, which covers about 90,000 square kilometers along the southern and western coasts of South Africa. It's designated as a UNESCO World Heritage Site due to its exceptional floral diversity of about 9,000 plant species, of which nearly 70% are endemic. It's a Mediterranean biome with a high degree of fire dependence and tolerance among the vegetation types. So to better understand links between fire, climate, vegetation, and people over millennia in the Fynbos, we're studying Islandvlei, which is approximately shown here by this white star. This is a coastal lake located at an ecotone between the Fynbos biome and small patches of the humid, fire-adverse Afrotemperate forest. So a little more on the site. It's the westernmost of three lakes that are located in the wilderness embayment, and today experiences high mean annual precipitation of about 900 to 1,000 millimeters that's distributed throughout the year as part of the year-round rainfall zone, which you can see in this map with the climatographs. So you can see that in contrast, most of central and eastern South Africa receives summertime precipitation while the western coast receives winter precipitation. And these zones are influenced by different moisture sources, where the year-round rainfall zone receives both tropical and temperate moisture. The vegetation at Islandvlei is aquatic, semi-aquatic, and terrestrial. And generally, Afrotemperate forest patches are found in valleys with higher soil moisture, while the Fynbos and coastal thicket vegetation is found occupying the coastal lowlands and surrounding dunes. So at the lake itself, the maximum water depth is about 6.5 meters, and the lake surface area is 1.38 square kilometers. The sediment core used in this study is shown by the white star and was taken in 2013 by Dr. Torsten Haberzettel and colleagues as part of the Regional Archives for Integrated Investigations project. This core was taken in about 6 meters of water depth and about 400 meters from the modern shoreline. So to date, this core has been analyzed for grain size and geochemical data, diatoms, pollen, and microcharcoal. And as part of my dissertation research, I'm working on a high-resolution macrocharcoal fire history reconstruction. And the work that Sandra and Nathan have done are working on black carbon data that we'll present here today. So in total, a composite core was put together of about 30.5 meters and spanning about 8,900 years. The full core was radiocarbon dated with 24 dates to produce a well-constrained age depth model. But for this research and my dissertation research, we're really focused on the top about 4,200 years, which is 9.6 meters of core. And this section has a sedimentation rate ranging from 4.5 millimeters per year to 1.3 millimeters per year, and generally slowing towards the present. So just to introduce some of my fire history reconstruction a little bit, I'm working with about 0.5 centimeter sampling intervals from the core, which has been about 2-year time steps between samples. And this is really neat because we can think about fire history in the deeper past on ecological timescales. And so I'm doing macro charcoal using 2 different size classes, so 250 micron and 125 micron size classes, which are approximate proxies for a relatively more local or more regional burning event. And so just to share a little bit of data here from about 4,250 years to 2,600 years before present. The gray bars are charcoal influx, and here it's just a sum of those 2 different size classes in the units of number of particles per centimeter squared per year. So you can see the influx is variable with periods of relatively higher or lower influx, as well as some individual high influx events that stand out. And now I've added a background trend to this data. So positive deviations above this background trend are interpreted as peak magnitude events, which can also be shown like this here, where these units are the number of particles per centimeter squared per peak and can be used to detect the relative size of the event as inferred from greater charcoal influx into the system relative to the background charcoal. And finally, we can look at fire frequency, which is the number of events per 100 years. And this output does adjust a little bit over time, but as I add more data, but we can still interpret that there's been variability in the frequency of fire over this time period, with some periods of relatively higher fire activity, which could be as much as 1 fire every 13 to 20 years or so, and then lower fire frequency periods as about 1 fire every 33 to 50 years or so. So in general, we're kind of seeing that towards the present, we have a bit of a decreasing trend in charcoal influx, peak magnitudes, and fire frequency. And with that, I'm going to stop sharing.
Thank you very much for this wonderful introduction to the fire history at Elandfley, Stella. So now in the second part, Nathan will walk us through all the method steps that were used at Elandfley Lake to establish a new black carbon record. Nathan is an assistant research professor in the Division of Hydrologic Sciences at the Desert Research Institute in Reno, Nevada. He completed his undergrad degree in geology and biology at Brown University in 2012, and then he did his master, his PhD, and also his postdoc at the University of Nevada, Reno, and the Desert Research Institute. Nathan's research focus is on developing paleoclimate records on centennial to millennial timescales, and lately he has been focusing on black carbon and biomass burning tracers. He works primarily with polar ice cores and just returned from Greenland, but he has also worked with alpine ice patches, lake sediments, and tree rings. Nathan enjoys spending time in the lab developing new methods and data sets, but he also loves field work to collect new samples. And in his free time, he enjoys hiking with his Stella. Nathan will now walk us through a method for measuring black carbon in lake sediment cores using a single particle soot photometer. And this SP2 instrumentation was initially designed for atmospheric measurements, but it has also been used extensively for ice core black carbon measurements and recently implemented for measurements in lake sediments. And with that, Nathan, I would like to give you the floor.
Cool. Thanks, Sandra. And thanks, everybody, for tuning in today. So I'm going to go through kind of our lab tour of the process for preparing samples for black carbon, sediment samples for black carbon analysis, but I just wanted to do 2 quick slides on black carbon background, just so we all on the same page. And so hope everybody can see that, but black carbon, like charcoal, is a proxy for fire. But the main difference being that black carbon is really, really tiny particles. So they're submicron soot particles formed by incomplete combustion. And so this image on the right here, oops, if you can see it. So these big black blobs in this image are actually sulfate aerosols, and the black carbon aerosols are actually these little tiny chains attached to them. So you can see some detail on the chain and individual black carbon particles on the right. And so they're on the scale of 50 to 100 nanometers. Typically, they can be quite large depending on how close you are to the burning source, so up to about a micron. But in general, we're still talking 2 to 3 orders of magnitude smaller than a charcoal particle. And so the other difference between black carbon and charcoal is that while charcoal retains some of that morphology from the original burnt woody material or plant material, black carbon doesn't have any morphology left, so it's a condensed soot product. And so that's black carbon, quick version of black carbon. And then black carbon in the atmosphere, just one big difference, because black carbon is so much smaller than charcoal, is that it can be transported long distance through the atmosphere. So here's some images of big smoke plumes coming off of Southern California. And then just, of course, black carbon has also been emitted from industrial pollution, from dirty coal burning primarily. But on the timescales for the lake we're talking about today, mainly natural forest fires are going to be the main sources. And so I've got a little video here now. We'll see if it works. So the first part I'm going to narrate over, and then the second part, there's actually sound in the video. So we'll see how well it works here. If it doesn't work, please put something in the chat. And so I'm going to go to this movie, share. And this is just going to be the movie playing for everybody. Play. There we go. Working? Okay. And so when we start with a like sediment core, you start with a core coming out in a tube normally, which has been halved. And so these are the samples that have been subsampled from the actual lake sediment core. These are U channels. And so it's just a subsample of the full sediment core. So this is what we start with, is the actual sediment itself in the lab and these U channels. And then Sandra and Stella worked on subsampling these U channels into about 3 to 5 centimeter resolution samples. So they were actually just taking the sediment out of those U channels, putting it in Ziploc baggies, and then shipping it back from Salt Lake City to Reno, Nevada. And so when they arrive at our lab, typically our sediment samples look like this. They're kind of muddy, they're in Ziploc bags and they're wet. So the first step is to dry them. And so we want to work with a dry sediment so we can constrain how much black carbon there is relative to the dry sediment in there. We don't want water to mess up we don't want to consider water when we're considering black carbon concentration in a lake sediment sample. And so we can see when these sediment samples arrive, they're often quite sticky and gooey. And so we first take them out of a Ziploc baggie and put them in these little aluminum trays that we will then put in the oven to dry at a gentle temperature for up to at least 24 hours, usually, depending on the sediment. So you can see how sticky and hard it is to work with. So when they're dry, they're a lot easier to work with, the samples, and then you don't have the water weight as well. And so I take the sample, try to be as clean as possible with it, wear the gloves. There we go, got our sample out of our bag, and step 1 is to dry it. I want to thank Sandra for helping me put this video together. It's a lot better than having me try to carry my laptop through the lab and show you all of this step by step. So there's the samples drying in the oven. Leave it in there for about 24 hours. For these samples, we are drying them between 40 and 50 degrees C, so it's a nice gentle drying. We don't want to char the material that's in them. And then we come back to the oven once the sample is dry, we pull it out, and there we go. We've got our dried sediment. It doesn't look that much different, but it's a lot easier to work with. And so we just put it into a vial because it's really easy to drop it off of those aluminum trays. And then the next sequence of steps I'm going to show you is kind of related to the extraction of black carbon out of the sample. So you can imagine there's a lot of different things in the sediment sample, and most of it isn't black carbon. And so we want to homogenize and grind up the sample so the black carbon can be accessed by when we resuspend this sediment in liquid for the analysis. And so once we've got the clump of sediment out, we take it over to the planetary ball mill and actually grind it up. And so this is a homogenization step. This is important to free up the black carbon from whatever else is in the sample, and as well as to make sure the sample itself is uniform. So we don't want to have any bias if we take a piece of sediment from this sample that has like a nugget of black carbon in it for example. it would give us an artificially high concentration. So we want to make sure the sample is as homogeneous as possible. So we put it in this agate bowl, add some agate balls to it, and then put it in this machine, which is called the planetary ball mill. And so it's called a planetary ball mill because it spins the sample while the sample spins in a circle, kind of like a planet in an orbit. And then those balls on the inside of that cup smash the sample into a dust, basically. It really homogenizes it and makes it easy to work with, and make sure that wherever we take our sample from, we get a uniform number. And so we let it run anywhere between 5 and 10 minutes, depending on the sample. We'll pull it out, and you can see the difference now as soon as I get it out. And so you can see that sample that was once that cohesive kind of chunk is now a nice fine powder. And so it's really easy to work with. The black carbon can kind of free itself from the whatever else is bound to it in the sample. And then the next series of steps is resuspending the sediment sample in aqueous solution. So the instrument we're using, Sandra mentioned, is the single particle spectrometer, which we haven't gotten to yet, but it was an instrument initially designed for atmospheric black carbon measurements. So it likes the sample stream it takes is an aerosol. And so in order to get an aerosol out of a liquid sample, which the instrument was first adapted to ice core research. So you're taking ice core melt water and using a nebulizer to aerosolize it into a gas, which then the instrument can measure. And then for the sediment cores, we have to do the additional step of adding, resuspending the sediment in liquid, extracting the black carbon into the liquid, and then taking the liquid through the nebulizer to make the dry aerosol to the instrument, which I'll show you here in a minute. But this part of the sample preparation process is a lot of weighing and a lot of tedious weighing. So we're putting about 50 milligrams of sediment out of that ground sample into the 50 ml vial to start with. And you can see me using that tiny little scoop there and adding sediment until we get to about 50 milligrams. And we're weighing it to keep track of the dilution. So how much sediment we add versus how much water we add. So that is controlling how much black carbon ends up in our sample eventually. And so it looks quite tedious, but a lot of science is tedious. And once you actually get comfortable with this process, you can actually do a full set of samples in maybe 3 hours, 2 to 3 hours. And we run about 60 samples a day for black carbon. So it's a pretty efficient way of measuring black carbon like sediment samples. So you can see me there. I like to weigh the vial twice just to make sure I get a robust measurement and make sure the scale's not acting up because of static electricity or wind drafts or anything like that. And so keeping track of what samples what. And so now we have our dry sediment in that vial. And as I mentioned, we need an aqueous sample. So we need it to be in water to run it on the SP2 via the nebulizer. And so the next step here is going to be creating the sediment dilution. So we add 50 mils of water to the sample. There we go. We got the dry sample in the bottom of the vial. See that little pellet of sediment. And then we add 50 mils of water. Just it's ultra pure water, so it's black carbon free. So we know we're not adding any black carbon. And then you can see how that fine sediment immediately resuspends in water. And then the idea with the next few steps is to mobilize the black carbon into solution so it stays there while the sediment can actually settle out, because the sediment density is much heavier than black carbon and much larger particles overall. And so the idea is that through sonication and shaking, which you'll see here in a second, the black carbon ends up in the water portion of the sample, and then the sediment ends up in that pellet at the bottom, and then we can pull off the water with the black carbon in it to actually analyze. And so again, we weigh the sample so we have a really precise control on the dilution. So we know exactly how much water there is relative to sediment. And you can notice there's a lot of weighing in this process. At least there's no nasty chemicals. That's what I like about it. It's pretty safe from a chemical standpoint. All right. And we continue to weigh. And then we have a worksheet that keeps track of all the dilutions and weights. So we know exactly how much sediment, how much water there is. Then we calculate the dilution that's corrected for. Here we go, shake, sonicate, shake, and sonicate. And so sonication uses ultrasonic sound of waves to break up particles and matter. And so by putting these samples in this water bath. The water bath then gets hit with sonic waves out of the instrument, and that's breaking up clumps of particles and helping mobilize the black carbon into solution. It makes a really, if you haven't used one before, it makes kind of an awful noise. It's this high-pitched, crackly noise. And so it's actually helping break up the sediment in there. And then I also shake them because you can see that sediment immediately clumps to the bottom unless you keep it up in suspension. And so this is helping mobilize the black carbon from the sediment with all of these steps here. And you can see how dirty those samples look. That's one thing to note here. As you'll see later on, the sediment tends to settle out quite a bit. But at this point, it's kind of it's a pretty dirty looking sample that we're working with. Sandra made this nice slow motion editing here to make it look very dramatic. And then, yep, more shaking, more sonication. Yeah, I have no questions in the chat yet, I assume. That's okay. All right, and then this has been sonicating now for about 15 minutes with a couple of shaking breaks in the middle where I take them out and shake it to make sure the sonication is actually acting. The sediment's actually in the water that's being sonicated and not just in a little pellet at the bottom. And then we put the samples on their side in a little Tupperware, little plastic container, and walk it down to a different lab where we put it on a shaker table overnight, which you'll see here in just a second. And so this is a full round of samples. This is a 63. This is a long day of analysis in the lab once they're done. So it's 3 racks of samples, so about 63 samples, which I guess if you started around 7 in the morning is usually done around 7 or 8 at night, depending on how smoothly everything runs. And so we don't have a shaker table in our lab, so we borrow one. So we walk through the halls of the Desert Research Institute, and there's There'd be a nice view of Reno out that window to the right. And then we take samples down to the shaker table, which just shakes them for 16 hours, because that's not something anybody wants to do manually. And so 16 hours on the shaker table and start, and off it goes. And so this is continuing that process of getting the black carbon into the water while leaving the sediment behind. And so these shake overnight. So then you come in the next morning, take them off, and walk them back down to the lab, our lab, and then we let them sit in the refrigerator for 24 hours overnight. And during that period, pause this real quick, during that 24 hours, the idea is that the sediment, which is a higher density than black carbon, settles out to the bottom in that pellet, and the black carbon actually stays in the water solution. So when we pull the water out of solution, we pull the black carbon with it. And so I'm going to try, this next part of the movie actually has sound. So I'm going to see if I can get that to work with sound, share sound. And so we'll see here. Okay. So this is the lab setup for the auto sampler to introduce the samples to the SP2. So on the auto sampler here, we can hold 4 vial racks. And so the rightmost 3 are the actual samples that you just saw us prepare. These are ones that Sandra's been working on. So you can see they have a nice little bit of sediment is settled out, but the black carbon is so small that it stays suspended in the water for much longer than the sediments. And then the general setup is you got the samples on the right, and then our standards here on the left. So at the beginning of each day, we run a set of calibration standards, and then we also run those throughout the day, every hour or so, to make sure the instrument response is staying about the same. And those standards are made from synthetic black carbon-like materials. Okay. So this is just to help you guys see how we make our standards and how they're diluted. So we start with this really concentrated black carbon solution. It's an industrial ink, so we know it's really stable in solution, so it's really good material for a standard. We dilute that down about 1 to, I didn't write it down on this, about 1 to 1,000, I think, for the first dilution, and we end up with a solution that looks still quite dirty, but not nearly as dirty as the original soup. And then from that is what we actually make our individual standards we run throughout the day on. And so by the time we've diluted those down, you can't even tell there's a black carbon in the samples, but we're calibrating as standards, sorry. We run 5 of these every day, and it's a concentration ranging from about 10 to 150 parts per billion. And we change those concentrations depending on the samples, if they're really concentrated or much cleaner. So that's just to show you how the black carbon is actually in those samples. The standards, it's just really hard to see. So that just look clear when they're over here because they've been diluted down. But basically we start with a black carbon-like solution here and do 3 steps of dilution so we get down to our final concentrations and our standards. And that's what we end up running to calibrate the instrument each day. And so the way the auto sample works is we just pump the water directly out of the sample over here to this valve. And this valve is set up so that the sample passes through one set of filters. There's a 20 micron and a 10 micron filter before it continues on. And if you notice, there's 2 sets of filters here. So it's set up that while one set of filters is being used for the sample, the other set is back flushing. So we switch between the 2 pairs of filters throughout the day so they don't get too clogged up. So after the water sample goes through the filters, it's pumped by our peristaltic pumps here, first to a debubbler, which just takes out the air. For example, right now. So this is the debubbler. And so when the auto sampler moves between samples, it puts air bubbles into the line. And so just to remove the air bubbles, we pump water and air in the bottom, and we take water out the top. And then because there's so much black carbon in these lake sediment samples that we have to pump in another dilution. So we're just pumping in DI water, which doesn't have any black carbon on it, on this precision pump. And so that dilution is going into one side of the T, the sample's coming to the other, and so we're diluting by a factor of about 10 to 1 for these samples. And then we pump that sample to the apex here, which is our nebulizer. And so that takes the water sample, mixes it with compressed gas, and shoots it into a spray chamber and turns basically the liquid sample into an aerosol, a dry aerosol. And then the dry aerosol comes out the apex through this tubing and goes to the SP2. And so the SP2 we're actually using is in the other room, but what SP2 looks like, it's just a nice metal box, sensors in there. And so that dry aerosol that we've generated from the water is getting sprayed into a laser that's going back and forth. And the black carbon particles, as they pass through the laser, are glowing. We're measuring the glow of those particles. So here's the actual data output up here. And so you can see, let me get a pointer. We just ran the quality control standard. These are the samples down here. And then the glowing of each particle is represented here by the peak height. So this is actually, these peaks is the photomultiplier tubes measuring the light emitted as each individual particle of black carbon goes through the laser. And then after this day is run, we'll take this data off the instrument itself and process it to get final black carbon concentrations. But that is the lab procedure there in a 16-minute nutshell. And then my last slide here is just our data. Final data set, well, preliminary data set from Lake Evangelie in South Africa. And so after you've processed and run all the samples, you stitch them together into a time series. You put it on the age scale that Stella showed earlier. And so for the black carbon samples, as I mentioned, I think they were sampled at 3 to 5 centimeter resolution, so a little lower resolution than her high resolution charcoal measurements. So about 18 to 20 years in time. And then you can see over the past 4,000 years at this site, similar to the charcoal, the black carbon deposition is also, or concentration in this case, has been decreasing towards modern. And so I just finished processing this the other day, so we haven't had much time to think about it, but there's a black carbon data set from the southern tip of South Africa. Yeah, and then I think that's all I have, unless there's questions and I can pull up some more details.
Thank you so much, Nathan. That was very interesting. I think we already have a first question in the chat. Can you see it?
How do you identify whether black carbon is present in the core or not? During initial assessment, just after cutting the core? So normally we just assume there's going to be some, at least, black carbon in the ice core. It's atmospherically deposited all over the world, but it's really widely distributed in the atmosphere. And so for most cases, there's going to be at least a little black carbon in the record. But that's a good question, because normally before we run a full sediment core, we'll do some spot measurements to make sure we're going to see something. That way we don't waste all of that effort and time to prepare an entire record just to find out. It's not going to be great. But in general, we haven't had a lake without any black carbon yet. We've had some with really, really low concentrations, but between the sensitivity of the instrument and the ubiquity of black carbon in the atmosphere, we usually think there's at least going to be some black carbon. All right, another question coming in. Are there different subfractions of black carbon of scientific significance, like wood source and size fractions? And so that's something, in general, was not yet. We haven't gotten that far yet in black carbon research. We're not sure if we can. So as I showed you those pictures of those black carbon little soot particles that are about 100 microns in diameter. And so those are like condensated out of the soot plume. So it's not actually anything representative of what was burning initially. And so one of the main factors that we think, instead of source emissions or the source material, we think a lot of what ends up in the black carbon record at a site is based on atmospheric transport, because those particles are so susceptible to different partitioning in the atmosphere and whatnot. And so we don't think there's much of a source signature preserved in black carbon once it's deposited, but that's certainly a further area of study is something like looking at size distributions or specific, because it isn't a single particle soot photometer is a single particle instrument. So it detects every single particle and there's some unique characteristics about it, which is an overwhelming amount of data that maybe would hold some answers to that, but in general, we don't think of it that way at this point. How do we differentiate between natural and anthropogenically derived black carbon in sediments? That's a good question too. Generally by time period. And so anything kind of after the Industrial Revolution between 1800 and 1850, we would be suspect to interpret that solely as a natural source. In some lakes, it's more obvious than others. You can actually really see industrial pollution. It's like a 3 to 5-fold increase in black carbon concentration. And then, you know, you're dealing with anthropogenic pollution. But in general, we kind of hedge our interpretations at that time for lake sediments to make sure we're not saying something about natural fire when it's actually human caused. Maddie, is your hand raised?
Thanks, Nathan. Yeah, I know there's a lot of great science questions, but I just kind of have more of a person question for you guys. So Sandra, Nathan, and Stella, you all work in the same lab together, right? And I'm wondering.
Sandra and I are at the same lab, and Stella's just 8 hours down the road in Utah.
Okay, so how do you guys work together? Is it all in the same project? Do you trade? What stages do each of you come in at?
Maybe I'm going to cut in here. So this collaboration kind of started because Stella presented her wonderful macro charcoal data at the last HU meeting. And so I immediately reached out and I was like, hey, Stella, do you just by any chance have some leftover sediments that you can share with us to try a fairly new method? And she was kind enough to start this collaboration with us. And then Nathan kicked in because he's the actual brain behind this method. I would be completely lost without him. So that's kind of like how we got to work together.
Thanks. Yeah, that's great to know. It's nice to know how these collaborations come about. I appreciate it.
Yeah, I think there's a question for Stella in the chat now too.
Yeah, thanks, Bill. So he says, I got the impression that the biggest fires tended to occur when the fire frequency had been relatively low. Is that right? And it's kind of, in some cases, what it's looking like right now. I mean, that could be something like we have more fuel building up when you don't have as many frequent fires. And so over time, when you have a higher fuel load and then the fire conditions are right, you might get some of these kind of mega fires, which, you know, is something we think we're seeing here in the Western US. I've been looking at this charcoal data in relationship with some published pollen data that's out there from the same core, and kind of watching how, like, you'll see fame boss vegetation starting to decrease when you might have more afro-temperate forest, vegetation increasing during periods lacking fire. So there's definitely some relationships between like the vegetation type and the fire frequency. And so, yeah, it'll be interesting to see how that kind of continues to go as we develop more fire frequency records, but it's showing a lot of variability where like the modern ecological records are kind of suggesting like fires every 10 to 13 years or something. And so it'll be interesting to see if that really holds true in the past.
I'll take a question here from Torsten. So what is the advantage of black carbon compared to charcoal? And so, yeah, I guess there's definitely pros and cons of each, but what really differentiates black carbon from charcoal is its long range transport. And so it can travel so long in the atmosphere that you can get really regional records from a single site. So ice core records from Greenland, for example, are representative of burning all across boreal forest regions. And so you get a really big picture image of what's going on with burning across the region, not just locally in the watershed. And that's one of the big advantages of trying to do that in lake sediments, where you're typically just doing charcoal. So you need a lot of local records of charcoal to get an idea of the regional picture. Whereas if you pick the right lake and the right depositional setting for black carbon, presumably you'll get a more regional picture from that record. That makes sense. And then, yeah, and then we wanted to adapt this method from ice cores to lake sediments to begin with. And because ice cores are limited to polar regions generally, or high alpine regions. And so by being able to develop black carbon records from lakes, we could have a lot more geographic coverage of black carbon emissions and deposition. All right. Yeah, another question here. Are there any post-depositional changes in black carbon and sediments once they sink in lakes? That's a very good question. And so on the timescales we're talking about here, as far as deposition, how the mechanism works from deposition from surface of the lake to settling with the sediments, that's hasn't been well constrained yet. So that's definitely something we're focusing on coming up. But as far as black carbon preservation and whatnot, like is it subject to any of the bioturbation or any kind of microbes eating it? We're not sure yet. Maybe on really long time scales, there might be some sort of degradation as it sits in the bottom of the lake for thousands and thousands and tens of thousands of years in some cases. But in general, on the time scales we're looking at, black carbon is thought to be quite recalcitrant. So it's pretty resistant to breaking down. It's pretty resistant to being eaten by bugs. And so we don't usually consider too many post-depositional changes for a black carbon record. And then can we really predict the range of black carbon transport and its provenance or sources within a catchment? Yes, I think we can actually do a decent job of modeling. There's some really sophisticated models. We've been working with modelers to interpret black carbon records, like using the GEOS-Chem atmospheric transport model, which does a really, really good job of actually simulating the atmospheric chemistry, the scavenging processes out of the atmosphere. It's a really detailed model, so you can only run really thin time slices with it because it's so computationally intensive, but it does a very good job of simulating how black carbon emissions from a certain source region would end up at a receptor location. Yeah, hope that helps answer that one.
So I don't see any question at the moment. So I have actually one for Stella. So now we talked a lot about how black carbon has like huge source areas, and you are working on macro charcoal. So what do you think is the catchment for your macro charcoal? Like what kind of area of fires does it cover for inland?
That's a good question. Yeah, it's something that there's increasingly been some papers coming out kind of trying to test some of the assumptions as far as like our 125 microns, our regional source area, and 250 microns, our local source area. Is that true? To what extent? What are the factors? And it really seems like this needs to be calibrated kind of for each lake individually based upon its own like catchment and depositional setting and prevailing winds and all that stuff. So people throw out some estimates, but for this particular site, I don't actually know at this point what my size classes mean, which is why I kind of just add them together and say this is sort of the general fire story. But I'm hoping to calibrate this a little bit better. I'm going to take some freeze cores this fall that are preserving like modern or historical fires. So if you know, like say there was a big fire in the year 2000 or something, and we have the data from that fire, we know how much area was burned or how destructive it was or any of those metrics. My goal is to try to calibrate what that known fire looks like in Island Blaze depositional record. And so hopefully that will provide a little bit of kind of calibration on what these fire events maybe mean in the deeper past compared to what they look like in the historical record. But yeah, hope that answers it.
Yeah, it does. Thank you so much. Do we have any other questions from the audience at this point? Nathan, I think you have a request to share your email with one of the participants. I have actually another question for Nathan. So this lake in South Africa is like really located at the coast. And so we can assume that we really like, because we have the ocean around, that we don't have a lot of local influences. But I know that Nathan has measured a number of other lakes in North America and one from South America too. So do you think like in some areas, the local contributions to black carbon are sort of overshading the long distance signal, or?
Yeah, so a lake sediment record, I think for black carbon, you need to really carefully consider the size of the lake versus the size of the catchment since atmospheric transport predominates for black carbon. So in a catchment where you have a huge lake with very little catchment area, most of the deposition we presume would be atmospherically deposited onto the lake, and therefore you'd get a good atmospheric record. But on the kind of other hand of that, if you have a tiny little lake with a huge catchment area with lots of water flowing into it, you would think that kind of more terrestrial sources and inflows would dominate the signal. So it's something you got to consider when picking a lake for a black carbon study is what's going to be the dominant contributor to the black carbon signal. Is it going to be atmospheric deposition with a small catchment, or is it going to be a lot more complicated of a story where you have some atmospheric deposition, but a lot more input from stream flows or surface movement of the black carbon throughout the watershed? So that does definitely complicate lake sediment black carbon records that we've been working with, where some seem to make a lot of sense. Those are the smaller lakes with a small catchment, and others are a lot more complicated and harder to interpret, which are generally these ones with a really, really large catchment area where the atmospheric signal kind of gets overwhelmed. That's a good question.
I was just going to answer the question about the material used for dating in the sediment core. It was bulk organic matter and some wood samples for the chronology.
Do we have other questions? If not, then I would like to thank Nathan and Stella again for presenting today. Please give them a last virtual applause. And also thank you all for joining us today and learning about black carbon methods in lake sediments.
Yeah, thanks.
And as a reminder, if you haven't signed up yet for the Pages Early Career Network, please register to receive information about many different networking meetings with other Pages Early Career researchers. And stay tuned for our next Pages North America Show and Tell event that will be announced soon. And with that, I would like to close and thank you all again very much for taking the time to join me, Nathan, and Stella today. Enjoy the rest of your day.
Sure. All right. Bye.